Welcome to Regional Heritability Atlas, a comprehensive web database on complex traits in UK Biobank. This web database is based on the research result that will be published shortly. It contains in-depth analysis result of 497 complex traits in the UK Biobank. In this video, we will go through a tutorial on this web application. You will learn how to use this website step by step. Firstly, you are going to learn how to utilize and interpret the phenotype-specific report. Click the By Phenotype menu button. This page shows a table of 497 phenotypes included in our study. You can sort the phenotypes according to a criterion you prefer. There are various criteria such as heritability, p-value of it, sex, LD score regression intercept, and prevalence, etc. As an example, let's sort by p-value of heritability. You can see that anthropometric phenotypes occupy most of the phenotypes having significant p-values. Now suppose that you are a researcher interested in the genetic factors of the body mass index. Then you can click the body mass index here. This page offers researchers a phenotype-specific analysis report. You can check the detailed information such as H2, LDSC intercept, prevalence, level of polygenicity. Here, you can see that the scaled level of polygenicity differs by the scale of segmentation we choose. Scroll down, and at the bottom of the website, you will find various graphical representation of analysis. This graphic is an interactive version of the plot included in the paper. First, let's see Landscape tab. The segments are split from genome by various scales. They are colored to indicate to which chromosome they belong. Let's zoom in here as a demonstration. If you hover your mouse on any segment, you can see how much variance this regional segment explains. For example, this segment, which is on chromosome 16, ranging from 0 megabasepair to 32 megabasepair, is responsible for 2.37% of total heritability. On the contrary, this adjacent segment, ranging from 32 megabasepair to 64 megabasepair, explains only 0.8% for total heritability. You can see that the boundary between segments are preserved quite well, down to finer segmentation scale, 8 megabasepair, since our segmentation method is highly robust. And for chromosome 6, you can see that a segment takes up large amount of heritability. If you try to identify the responsible segment on finer scale, you may finally assure that this was due to the highly pleiotropic region, called major histocompatibility complex. Additionally, if you click a segment, you can only check values of it. This result may be used as a reference data to prioritize regions for further fine mapping study. On the other tabs, you can check correlation and ANOVA results. Let's see correlation. For example, phenotypes that are correlated with BMI are as follows. If there is a phenotype that interests you, you can just click on it, then you will be moved to corresponding phenotype-specific report. In ANOVA tab, the unevenness of the regional heritability, observed in various segment scales is shown. For example, for body mass index, this figure says that the finer the segmentation scale becomes, the more significant unevenness of the heritability from segments becomes. In other words, for BMI, we have to split segments on finer scale, to check the unevenness of distribution of heritability. Until now, you learned how to utilize and interpret the phenotype-specific report. From now on, you will learn what kind of information can be retrieved from this All Phenotypes menu. In the first submenu, Overview, you can look at philosophical questions and answers on our research. In the second submenu, Polygenicity. You can compare level of polygenicity and heritability among traits. For example, in the upper right corner there are anthropometric traits such as weight, impedance of arm, BMI, etc. Under them, there are hematological traits such as mean platelet volume and red blood cell distribution, etc. It shows that, although anthropometric traits and hematological traits have similar heritability, anthropometric have higher level of polygenicity than hematological traits. In the third submenu, Browse by region, you can find which genomic segments are pleiotropic. Pleiotropic region abnormally explains large amount of variance for multiple traits. For example, you can see region around MHC as very pleiotropic. If you click a phenotype, you can move to the phenotype-specific report page. In the next submenu, Phenotype Network, you can look at correlation network among traits included in our analysis. Phenotypes of similar genetic background are clumped together closely. For example, phenotypes such as neuroticism, mood swing, irritability clump together, implying that the phenotypes share similar genetic background. Also, interestingly, greener dots, representing high level of polygenicity, are located in center of the graph. Similarly, you can click a phenotype of interest and move to the phenotype-specific report page. Lastly, in submenu ANOVA result, you can check ANOVA result about unevenness of the regional heritability as explained previously. Additionally, in the More menu, you can check technical details, download raw data and source code, and see how to contact us. Thank you for watching this video. We hope that our web database help or accelerate other research's progress. This website was built as a part of research project at Genomics and Bioinformatics Laboratory at Seoul National University. If you have any question, feel free to contact us.